of energy.
Our Earth is running out of sources.
Lots of scientists are eager to find solutions.
This problem has to be solved.
In 2017, CGU Taiwan was formed.
Our project is started on recycling paper.
4 billion of trees are cutting down each year.
Lots of homeless animals.
They are seeking a place to live.
1 ton of paper needs 3.5 tons of wood.
However, 1 ton of paper can make 1 ton of reprocessed paper.
Do you think reprocessed paper is the best way to protect the earth?
No, there is still another problem inside reprocessing paper.
A large amount of wastewater.
A big amount of chemical agent has been used.
And water was polluted.
Water is health,
Water is nature.
and water is energy.
We have a dream.
We want to lose the amount of speed of amount of losing energy.
We want to reduce the pollution.
We want to have a healthier earth. Hello, ladies and gentlemen. We are CGU Taiwan iGym 2017. As you can see, our logo is ink off. Our team wants to do a reprocessed paper. However, there is a problem inside reprocessing paper. A big amount of chemical agent has been used. So, what our team want to do? Our team want to reduce the chemical agent. Our team want to secure the natural resources. Our team want to make a difference. So here are 2 questions I want to ask you guys. Why the reprocessed paper cause pollution? And in which process a chemical agent has been used? Since nobody knows the answer, let's start with the introduction.
So now let's start with knowing how the reprocessed paper is produced in industry. The recycling paper is firstly made into paper pulp. And this pulp is cleaned and screened by removing the trash. In the inking process, the ink is removed from the recycling paper by using chemical agent. Next, the pulp is then fiberized to create a stronger paper. After suspending and preparing stock, this pulp is pressed and dried, and finally the reprocessed paper is produced. So let's go back to talk about the reason of causing pollution. As I mentioned in the previous slide, there is a large amount of chemical agent used in the de-inking process. Consequently, a large amount of wastewater is produced. But now we have an idea to solve this problem. In brief, we are going to construct a genetically engineered plasmid, and this plasmid is transformed into yeast. And this yeast will secrete out the inking enzyme. Those helps to remove the ink. And this yeast will show its effect on a de-inking device that we designed. A light-induced enzymatic de-inking device. First, we are going to use a scanning scale to localize the inking part of the recycling paper. And the enzyme secreting yeast is sprayed on the recycling paper. And then we use the LED light to induce this yeast. So the yeast will produce the de-inking enzymes to remove the ink. To make this device, we need to attain a series of procedures. So our idea is to develop an enzymatic de-inking method instead of chemical method. So we are going to build a model and design a de-inking device. In dry lab, computer simulation is done to choose the enzyme those are best fit to our need. After choosing enzyme, we can start to construct the plasmid and enzyme secreting yeast. And finally, the marketing work will help to investigate whether our product is fit to our industry need or not, and also improve the current industrial de-inking method. So our team is divided into 5 different groups to fulfill this mission. So at first, we are going to design a de-inking device to build a small-scale de-inking process model and put the industrial line into the lab. Our engineering and model design group is going to fulfill this mission. As I mentioned before, we are going to design a light-induced enzymatic de-inking device. So this device consists of 3 points: camera, logic unit, and light output. So first, we are using camera to take picture of the reprocessed recycling paper, and this image will connect to the logic unit. So this logic unit is to analyze the de-inking part of the paper, and this logic unit will send a signal to LED light to control this light. So when this LED light receives the signal, it can induce the yeast to secrete out the de-inking enzyme. So by using this enzyme, we can specify the de-inking part so we can reduce the damage on the recycling paper, and also we can improve the quality of paper. After designing the device, the computer simulation is done to choose the enzyme. Because there is a large amount of organisms producing different kinds of enzyme. So how can we know which is the most suitable enzyme and how we can modify this protein sequence? So the information and modeling group is going to work on computer simulation to solve this problem. In modeling part, we will mainly test for the binding affinity of enzyme and ligand by using different kinds of modeling softwares. First, we are going to search for the templates of the similar proteins by using Swiss model. And these proteins, we need to download all this file from the protein data bank PDB. And before the protein docking, these proteins have to be fixed. And the ligand also have to be minimized energy so we can get a more accurate result. After preparing all the conditions, we can now move to our protein docking by using Autodock. This Autodock software is to simulate the binding between enzyme and ligand and calculate the binding affinity. So as a result, we can get the binding affinity ranking of each of the proteins and also visualize the docking situation by using PyMol. Finally, the biodevelopment group can construct the plasmid based on this docking result.
Okay, after our information and modeling groups, here comes to our wet lab. Our wet lab is done by our biodevelopment groups. They are constructing the plasmid and build the enzyme de-inking, secreting out yeast. Here's our biodevelopment groups, and let's start with introduce our plasmid. Here's the 5 plasmid we will build. The top two will be our light-induced plasmid, and the bottom is our 3 enzyme. The first enzyme is xylanase, second is glucanase, and the third is lipase. For xylanase and glucanase, they will cut off the paper fiber, so the ink will float in the water, so will come off. And for the lipase, it will bind to the ink and help the ink come off from the paper. As you can see, we choose 5 plasmid and also using different selection marker. Next, why are we choosing yeast as our modeling creature? As the animation goes on, you can see our yeast baby is eating all the plasmid. Yeast have a big tolerance of having huge amount of plasmid. First of all, why we choosing yeast? It's because we want to using the secrete out pathway. We don't want to purify the enzyme and spray on the paper. We want to spray the yeast directly on the paper, so it will secrete out the enzyme and working on the paper. Second, Yeast is well researched. It's not a brand new field for us, so it's easy for us to get started. Third, yeast is easy to grow, and actually it grows rapidly. It only takes 2 days for yeast to grow, instead of 1 week. Especially in this type of competition, time is money. Fourth, yeast is strong. You don't want to transform your plasmid into your modeling creature and just dying. It's awful. So we want to choose yeast. Next, why are we choosing light? First of all, it's eco-friendly. It's perfect fit for our project. Second, light can target specific place. So we can just zap the light on the inky part and let our yeast secrete out the enzyme and working directly on the inky part. Third, light is easy to control and actually it's easy to bend. A lot of machines have this kind of condition, just shine the light on the paper. Fourth, light can improve the quality of paper. Why? Because I mentioned before, xylonase and gluconase will cut off the paper fiber. So if we can use light to control where our enzyme wants to de-ink, in the later process, after we destroy the paper and rebuild the paper fiber, it will be more strong and we can create a good quality of paper. Why are we using enzyme? The same reason, it's eco-friendly. Second, it's produced by creatures. So instead of put a lot of chemical agent on the paper, we can just spray the yeast on and let the enzyme keep creating. Third, it can reduce the use of chemical agent. Chemical agent is harmful for environment. That is the main goal, why we're using enzyme to replace a chemical agent. Fourth, it can reduce the amount of wastewater. If we do a pretreatment on the paper, our enzyme already working on the paper fiber. So in a later process, we don't need to add a huge amount of water into it. So we can reduce the wastewater. To fulfill one of the criteria, improving biobricks, we are choosing 2012 Moonchus biobricks. You can see, this is a light-induced biobricks. It will produce 2 fusion protein, and we are using Tef1 promoter. It's a constitutive promoter in the yeast, so it will keep creating a fusion protein. You can see in the picture, these 2 fusion protein, the first one will be a 5B domain and DNA binding domain. The second one is, you can't quite see it, but it's PIF3 domain and activation domain. After the red light shot on our fusion protein, these 2 fusion protein will recruit together and express the downstream gene. Although I said in previous slide, light have a lot of advantage, but it have a big disadvantage that we don't want. It will reduce the amount of protein efficiency. That's why we want to improve this biobricks. So what did we do? First of all, we reverse the promoter and target gene. As you can see in the previous slide, the promoter is here and it's going this way. But we reverse it. Why are we doing this? We want our RNA polymerase bind and working in different direction, so it won't influence to each other. Second, we do an optimize our open reading frame. We change the amino acid to what yeast like, so hopefully it will produce the highly protein. What we want to see? Of course, the highly protein efficiency. After all our project was done and device was made, here comes to our marketing groups. Our marketing groups will shorten the distance between the industrial and our lab. They will tell people we are not just a bunch of people sitting in a lab. We are someone, we are doing something. And also, we are interviewing with a lot of experts about our project. So, in March, we went to visit Dr. Su. Dr. Su is a Zhongxin University's professor. He is an expert in enzyme de-inking. He gave us a lot of suggestions on how to combine the enzyme de-inking into industrial line. And next, we went to visit Dr. Zhang. Dr. Zhang is a professor from Tsinghua University. He is an expert in yeast-2 hybrid system. We use this system as our light control. So, because the yeast-2 hybrid system is a brand new field for us, he gave us a lot of suggestions about the design of experiment. After visiting all these experts, we went to Taoyuan High School. We went there to teach students how to use pipette, how to work in a lab, how to be a scientist, and the most important, what is IgM what is synthetic biology, and what it would do. Not just high school, we also went to elementary school. We can't teach them too much hard knowledge because they don't understand it. We design a lot of game, and through the game, they know how to make a reprocessed paper. And also, they know the idea of protecting Earth, protecting environment, and they know who are we. After visiting the school, the most important part, shorten the distance between industrial and lab, we went to visit company. The company we choose is Zhenlong. Zhenlong is one of Taiwan's biggest paper-making company. They show us the industrial line, and they also give us a suggestion about how our project can be more attractive to the industrial. And the last, 2 weeks ago, we went to Science Carnival. We attend this event. A lot of people come to our spot, so we can tell them who are we and what we are doing. To conclude, we are making a device.
We are solving problem.
We are making a bioenzyme de-inking device.
We are securing natural resources with no pollution.
Our device have a light-induced device. Our device have an engineered yeast. Our device can solve the industrial problem.
Our device reduce the use of chemical agent. Our device reduce a big amount of wastewater. Our device secure the natural resources. We will
lead it to a better world. Thank you. This is the end of our slide.
Thank you for Chang'an University's presentation. And any other questions?